Namaste students, Kulshwa Vidya. I, JV and Dr. Chandrakala, Assistant Professor at Vidyapeeth, Women's University, Jaipur. In today's session, we are going to discuss about the protein sorting through non-secretory pathway. As the protein are intrinsic signals that govern their transport and locate in the cell that are discovered by Gunther Global in 1999, for which he was given awarded Nobel Prize. The pathway of the protein sorting includes the secretory pathway and the non-secretory pathway. Secretory pathway begins in the endoplasmic reticulum. New synthesized protein excluded from the ribosomes are directed into endoplasmic membrane. Once the translocation across the endoplasmic reticulum and they are transported to the Golgi complex via a vesicle. So then these protein present within the membrane enclosed vesicles are mainly secreted outside the cell of an endocytosis. Some of them are targeted either to the plasma membrane or to the lysosome. Non-secretory pathway. Non-secretory pathway mainly includes the target of a protein as follows. Protein, that is the cytoplasm, mitochondria, peroxisome, and nucleus. So there are four locations inside a mitochondria to which the protein are translocated. They are mitochondrial matrix, inner membrane space, outer membrane. Okay. The mechanism of the targeting into a protein targeting into a matrix. The precursor protein synthesized on a cytosolic ribosomes are maintained in an unfold or a partially fold state by bonded chaperone only. Such as cytosolic, HSP70 or use of energy derived from ATP. The mitochondria are recognized by an import receptor composed of the TOM20 and TOM20. The import receptor subsequently transfer the precursor protein into an import channel into the outer membrane. These channels are composed of the TOM40 protein known as the general import force because all the known mitochondrial precursors proteins gain an access to the interior compartment of the mitochondrial fluid. In the case of the precursor, distinct from that of the mitochondrial matrix, the transfer through the outer membrane occurs simultaneously with the transfer through an inner membrane channel composed of TIM20 and TIM17 protein. The translocation into the matrix thus occurs as a contact site, okay, where the outer and the inner membrane are closely proximate. Soon after the N-terminal, the matrix targeting the sequence of a protein enters the mitochondrial matrix. It is removed by the protease that resides within the matrix. The emerging protein is also bound by the matrix, HSP70. A chaperone that is localized near the translocation channel in the inner mitochondrial membrane by the interaction with the transmembrane protein TIM44. So this binding stimulates the ATP hydrolyzed by the matrix of HSP70 and together with the TIM40 and HSP70 and thought to be a power translocation of the protein into the matrix. The final folding of many proteins require chaperones present in the mitochondrial matrix. The mechanism involves the three pathways, that is path A, path B, and path C. In the path A, in this pathway, the precursor protein contains N-terminal matrix targeting a sequence of the TOM20 by 22 import receptor. Thus, it transfers through a TOM40 general import force of the outer membrane and through pin 23 by 17 translocate the complex of the inner membrane. So here as the protein passes through the 23 by 17 channel, the hydrophobic stops the transfer, anchors the subsequent blocks the translocation of the C terminal across the inner membrane. So the membrane anchored the intermediate is then transferred laterally to the bilateral and the inner membrane. The second pathway is the pathway B. In this pathway, the precursor protein contains both the matrix targeting the sequence of the internal hydrophobic domain recognized by the inner membrane protein and is termed as OXA. So, after the completion of the transferase, the protein into the matrix targeted sequence is cleaved. So the cleaved protein is inserted into the inner membrane and is the process required the interaction with OX1. So the pathway third is the final pathway where the precursor of the protein from the insertion of the inner mitochondrial membrane is followed by a multiple path protein as the ATP and ADP antiporters. So here this protein lacks the N-terminal matrix targeting the sequence containing the multiple internal mitochondrial targeting sequence. After the internal sequence are recognized by the second import receptor composed of the outer membrane protein TOM70 and TOM20. So the protein are transferred to the second translocation complex to the inner membrane composed of the TIM20 and TIM80 and TIM54 proteins. These small ring proteins are guided by the imported protein precursors and followed by the complex inner membranes by binding the hydrophobic regions. The mechanism of the protein targeting into the intermediate space of the mitochondria is followed by the two pathways, that is again path A and path B. So in this pathway, the precursor of the protein carry two different N-terminals targeting sequence, both into which ultimately cleave. The matrix targeting the sequence is removed by the matrix protease and is the hydrophobic segments of the block completely by the translocation of the protein membrane. So then this precursor fuse to the laterally and the protease in the membrane cleave into a protein near the hydrophobic translation to the interspace. In the part B, the protein do not contain the N-terminus matrix targeting the sequence. So they are delivered directly into the intermembrane space via general import port without the involvement of the intermembrane translocation factor. The mechanism of the protein targeting into the outer membrane. Many of the proteins that are resides to the mitochondrial outside the membrane includes the TOM40 pores itself and the mitochondrial purine have a beta barrel structure. Such protein and are incorporated into the outer membrane by the first interaction with the general import pores of the TOM40. They are then transferred into the SAM complex composed of the three outer membrane proteins. One of these three proteins are SAM50 and which necessary for the insertion of the beta barrel protein. Finally, the protein is laterally transferred into the bilateral of the outer membrane. The peroxisomes are small organelles bounded by a single membrane. All enzymes found in the synthesized in the cytosols. That is example catalase, urease, oxidase. So the mechanism of the protein sorting in the peroxidase. So in this cytosol, the PTS1 binds to the receptor called EX5. It has the ability to switch from the monomeric forms to the oligomeric forms, which is then embedded in the complex protein called the PEX14 and the peroxisome membrane. The PEX14 adjusts itself according to the size of the PTS1 bearing the cargo molecules. The peroxidase zone impose the machinery translocation force protein across the membrane. The PEX5 recycles involved in the modification of the membrane bond PEX5s and a complex of the peroxisome of protein PEX10, 12 and 2 transfers the moiety to the PEX5. So the PEX1 and the PEX6 anchors to the peroxisome membrane by the PEX15 recognize the PEX5s and remove it from the oligomeric complex with PEX14 with the help of ATP hydrolase, thereby releasing it into the cytosol. After the removal of the cytosolic PEX5 is ready to carry out another cycle of binding the PTS1 bearing protein. The target sequence are not removed from the matrix. So the targeting of the protein into the nucleus. So all proteins found in the nucleus are synthesized in the cytoplasm. Example, the histone, DNA, polymerase, or the factors. So the transport required in the nuclear translocation signals here is the mainly consists of several residues rich in the amino acid near the C terminus of the protein. So the nucleus protein is the nuclear core complex are made up of three nucleophorein: structural nucleophorein, membrane nucleophorein, or the FC nucleophorein. Okay, so the mechanism of the protein sorting in the nucleus. It imports the protein containing the nuclear localized signal sequence required a nuclear transporter receptor and is known as importin. These pre-importins in the cytoplasm binds to the cargo protein, forming the import complex, cargo complex. The then the RAN interact to that of the GTPase, activating the protein. The RAN GDP travels back to the purine and forming a nucleoplasm, where the encounter specific guanine and nucleotide exchange the factors. The protein are responsible for near the every task of a cellular life, and each compartment of the cell functions mainly through the protein. So the protein sorting is inevitable phenomena. Thank you. This session is powered by 2.0 Jyoti Vidyapeeth Women's University, Jaipur. If you have any queries,